So if we look at kind of what makes our cells become cancerous, because cancer cells are our own cells. This is essentially our cells that have been warped, and so now they're not behaving properly. That's why it's so hard to attack and kill cancer, because in terms of what the cells look like and what they're composed of, they're our own cells. So it's harder for our immune system to target, hey, is this cell, one of ours but not doing its job, or is this one of ours and doing its job? Chemically, that's pretty difficult to do. That's why it's so difficult for us to treat cancer. That's why it's so difficult for our own bodies to fight the cancers. But the way in which this works is you typically have several genes that say don't be cancer. These are going to be called proto-oncogenes and tumor suppressor genes. And so these various genes are going to make sure that your cells listen to the appropriate checkpoints, they behave appropriate. But if you have some type of mutation, which could be caused by X-ray exposure or UV light exposure from the sun if you tan a lot, could be caused by carcinogens that are in smoke if you're a smoker, but there's lots of things that can lead to mutations. And if you mutate these tumor suppressor genes to where they break, they stop doing their job, you're now one step closer to cancer. If you mutate these proto-oncogenes and convert them into what's called oncogenes, that's the ones that say go ahead and be cancer effectively. So if we damage these proto-oncogenes as well and make them become oncogenes, if one cell gets all of those mutations, now you have cancer. So to make it clear, a single cell in your body has to have enough mutations that break all the tumor suppressor genes it has, of which there can be a couple, and all the proto-oncogenes it has, of which there can be a couple. So when it's left with these broken tumor suppressor genes and oncogenes, that cell has now become cancerous. And if that cell is not weeded out by our immune system promptly, it's going to start to divide uncontrollably, and you can now have cancer. This is why it's such a big deal if you have cancer that they'll say we have to get every last bit of it. So they don't just remove the parts that look like cancer, they try to remove all the tissues around it to make sure there's no stray cancer left, and oftentimes they still treat you with chemo, trying to make sure there's not some stray cancer cells somewhere so that they can try to take care of it. So mutation is going to be the key. Now this is also why it tends to be an older person's disease, because the longer you live, the more you're exposed to these different carcinogens or mutagens, things that can lead to this, like we said with cigarette smoke, certain chemicals like asbestos. There's a bunch of stuff that can make you more likely to have mutations, and as time goes on, you're more likely to get some of your cells get all these mutations necessary to become cancerous. That's why you worry about this when you're 50 or 60, but not as much when you're younger. You typically will not get cancers, most cancers, in your teens or your 20s. So if this occurs, at that point, we go through aggressively and we try to take care of it. Just on a side note, a lot of the treatments too, like chemotherapy, they function because cancer is so ravenous, because it's trying to grow so fast, it needs lots of energy and resources. So we basically put toxins in your body because the cancer, because it's taking in more stuff than almost all the other cells in your body, tends to ingest the toxin faster than your other cells. That's why we're able to kill off the cancer, but you also feel like crap because we are damaging your own cells, especially cells in your body that reproduce rapidly, like the inside tissue of your mouth or your hair follicles. They also use up a lot of resources, and so because they use up a lot of resources, they also take in more toxins. So this is why chemotherapy is kind of an interesting treatment. So you're poisoning the body, hoping you kill the cancer faster than you kill yourself. Now, there are some things, too, that are inherited mutations. So this is like BRCA1 and BRCA2. These are breast cancer genes that sometimes you can be born with. So while you're not normally going to be born with cancer, you can be born with a predisposition for cancer where some of these proto-oncogenes we talked about are already converted to oncogenes. And so it doesn't take as much to become a cancerous cell now because some of those kind of safety switches that we had in place are already flipped off. You can also have situations where viruses can give you oncogenes. So there are a couple viruses that they talk about. This is why HPV can lead to cervical cancer and some types of ovarian. And that's why they're trying to give the HPV vaccine, is because if you get that virus, it can insert an oncogene and make you predisposed to get that type of cancer. Now, apoptosis is kind of a cool idea. You've seen in the movies where there's this kind of self-destruct button. So if things go bad, we can ultimately get rid of the whole thing by having this controlled destructive phase. And so in our cells, we have something very much like that called apoptosis, where if we send a particular chemical signal from our immune cells typically into a cell, it triggers it to go through this process where it goes through suicide. And the way it does this is it starts to break itself down where it forms these little pieces that stick out, that are these little vesicle type regions. It cuts up its own insides, its own DNA, etc., using a variety of enzymes. And then as it forms these finger-like extensions, it packages itself up in these nice little packets. So the one big cell becomes a bunch of chewed up stuff in these small little vesicles that other cells nearby can then grab and digest to take care of. So this means that if there was something bad, it hopefully got destroyed by being cut up, and then the other cells can just ingest it, and they can cut it up with their own enzymes and even get a bit of energy from it to allow this process to function safely so we don't spread things. So if we have a cancerous cell, if we have a virus-infected cell, we can try to go through this process of killing it in a very safe way so we're not infecting other cells. There's also uses for apoptosis during development. You'll see that during early development we had webbed fingers, but then we use apoptosis to kind of gnaw away at those cells, and so as those cells disappear and get reabsorbed, we now end up with normal fingers that are not webbed. Although some people don't do it fully, and so you might see some people that have extra webbing because they didn't do a very thorough job of the apoptosis. But we can use apoptosis for kind of developmental sculpting of our form and shape, which is kind of cool. Where we had some structures, they went away, and when you wonder like how'd they go away, the answer, at least in some of those cases, is apoptosis of those cells to get rid of them. Now, stem cells is a somewhat controversial idea, but these are cells in our body that haven't picked a job yet. When your cells start to develop, they eventually become, say, a liver cell or a kidney cell or a muscle cell or a nerve cell. And once they do that, they're locked in. So if they reproduce, they'll produce a cell just like them. You know, if this would be kind of like if you guys picked a career and then you're stuck with that career for the rest of your life and all your kids are stuck with that career for the rest of their lives and your grandkids, etc., etc. This is how cells typically work, especially in animals. But there are some cells called stem cells that haven't picked a job yet, so if they're given the right signals, they could become any type of cells. You see these present early on in the development of organisms, back when they're still essentially like a zygote, when they're still like an early embryo, because those cells have not been given their ultimate job yet. You're just kind of a indeterminate ball of cells at that point. And so if we use these cells, the idea is that we could give them the proper signals and we could create whatever tissue we need, or if we could get just the right amount of signaling, we could potentially grow whole organs that we could then give to people to replace faulty organs or damaged organs that they may possess. So there's a lot of potential with stem cells that we could fix things like paralysis where the nerve cells are damaged. If we could make people more nerve cells, then we could fix that. Now, early on when they did a lot of this research, they would use embryos as their source of stem cells. And so because they were using embryos, typically from things like fertility treatments, where people would get a bunch of embryos, they'd implant a few, and then there were these embryos left over. And so they were like, let's use these and see if we can do this research to try to help people. But because some people didn't like the fact that we were using embryos for that, even though the embryos were just going to be destroyed typically anyway, so it wasn't really much of a change. But a lot of people didn't like this idea because they think, well, an embryo, even though it's this basically undifferentiated ball of cells, it has life, and so we don't want to harm it. And so nowadays, you'll see there's a lot more focus on working on adult stem cells, because we do have where there's many cells inside adults that can become many different type of cells, even if not necessarily all of them. So adult stem cells are not necessarily as perfect as embryonic ones, but they're at least getting to the point that we might still be able to use them, and it gets rid of this ethical issue. It also makes it easier because they're a perfect match, so we don't have to worry about trying to make sure that we pick an embryo that has the proper genetics so you won't have rejection if I put it inside you. We've talked before about this idea of there's carbohydrates and proteins embedded in the membrane that help identify who the cell is. And if you have the wrong identification, we attack it. And so we can bypass that if we can use people's own stem cells. So there's a lot more effort now into focusing on things like bone marrow. We've also found that there's stuff that we have in fat that's our stem cells that we can use. And so there's a lot of research in trying to use our own adult stem cells and perhaps give them a little bit of a push to be a little bit more complete of a stem cell. We're trying to help work to de-differentiate them, to make them go back to be a complete early stem cell. And so in this way we can grow whatever we need so we can still make someone spare parts. We call that therapeutic cloning, where we could try to produce someone tissues or organs, so that way we don't have people dying waiting for a liver or a kidney when we could just get one for them given 6, 8, 12 weeks, whatever it would take to grow one. We could then implant it in them and they could live. So this would allow a lot of people to survive for a lot longer in terms of lifespan when they get various diseases that damage their liver, their kidneys, and other organs that we have to have a replacement for. You can't survive without.